बंसिखा, मिस बंसिखा.
Hello.
मिस बंसिखा. Miss Bansikha. Okay, next participant Aishwarya Sharma. Next participant Aishwarya Sharma. Aishwarya Sharma, please start your presentation. Next participant Pankaj. Next participant Pankaj. Monali Nikesh.
So this is my topic of my.
Monali Nikesh. Yasika. Next participant Gitanjali. Next participant
Yes,
Gitanjali.
Yes, sir.
Please present your paper.
Sir, my screen is visible.
Yes, visible, very much visible.
Okay, thank you, sir. Good afternoon, everyone. My name is Gitanjali Shukla, currently pursuing MSc in Babasaheb Bhimrao Ambedkar University, and my topic of presentation is correlation of cell death and oxidative stress. As we see the topic, the first question which arises in our mind, which is what is cell death? So cell death is a process which occurs when a cell stop working, but before it stop working, it divided in more than 60 times. This process is called cell division or mitosis. The second question we thought about is what is oxidative stress? Uh, so the oxidative stress is an imbalance of production of ROS and its ROS and body's ability to neutralizing it. And the source and mechanism of oxidative stress. So the ROS is a source of oxidative stress, which is reactive oxygen species, which is highly reactive because it is an unpaired electron. It is also called an unstable molecule. What is the sources of reactive oxygen species? uh, and there are two types of sources, which is endogenous and exogenous sources. Endogenous sources which arise from within the cell, they are major components which are produced from mitochondria or endoplasmic reticulum. They are main generators of the endogenous ROS. Exogenous sources which arise from outside of the cell, these are environmental pollutant, radiation, cigarette smoke, certain foods, and drugs. As we can see in these pictures, on the left hand side, we can see the endogenous, which is UV rays, gamma irradiation, cigarette, and all. And on the right hand side, we can see endogenous sources, which is mitochondrial, electron, transport chain linkages. And on the upside of the slide, we can see antioxidant, vitamins, and coenzymes. Uh, it helps in high production of ROH, which leads to oxidation of proteins, DNA, and lipids, which causes cell death. The effects of ROS, there are both types of effects of ROS, which is beneficial and harmful. At low level, ROS shows beneficial effects, which is ROS can help regulate signaling and kill the bacteria, sperm captivation, fertilization, embryo development, regulatory mechanism, intracellular signaling, host defense against invading microbes. And the harmful effect at high level, ROS can damage DNA, protein, and lipid per oxidation within the cell membrane, oxidative damage to protein, mutation of DNA, activation of pro-cell death, and factors. There are two types of pathways which is triggered by oxidative stress. Is uncontrolled form of cell death, and which is controlled, for apoptosis, which is programmed form of cell death. The programmed cell mechanism follow two pathways, which is intrinsic and extrinsic pathway. The intrinsic pathway is inherited by intracellular causes such as DNA damage, mitochondrial dysfunctions of endoplasmic reticulum stress, and the extrinsic pathway is inherited by external signals that bind to specific cell surface receptors called death receptors. As we can see in the slide, in the diagram, the first stage of the programmed cell death mechanism is elimination of unwanted cell, and then cells started to shrink, and then nuclear started to fragment, and apoptotic bodies we can say in fourth stage, and at five stage phagocytic of apoptotic bodies. Oxidative stress can induce apoptosis, which is damage to DNA, other cellular components, activation of stress signaling pathway, disruption of mitochondrial functions. Mechanism of necrosis. Oxidative stress is an imbalance between reactive oxygen species and antioxidant defense, can trigger necrosis through various like mechanism, mitochondrial dysfunctions, endoplasmic reticulum stress, membrane damage. The process of necrosis always start to pathologically, then cell start to enlarge, then loss of membrane integrity, then leakage of content, then inflammation started and nuclear degeneration, and after that the cell is dead. Oxidative stress can lead to different types of diseases like neurodegenerative diseases, cancers, and uh, these are the references. Thank you, sir.
Thank you, Gitanjali.
Thank you, sir.
Next participant, Unnati. Next participant, Unnati.
Yes, sir.
Please present your paper.
Sir, is the screen visible?
Yeah, it's visible.
Good afternoon, everyone. Myself, Unnati, from Department of Zoology, Baba Saheb Bhimrao Ambedkar University, Lucknow. Today's my presentation topic is genetically modified organism and their impact on biodiversity. Introduction. Genetic modified organism, also known as living modified organism, transgenic animal, and genetic engineered organism. GMOs have many advantages and some disadvantages. Advantages are study any disease in any animal at the genetic level is possible. GMOs increase high yield in crops, reduce chemical uses in feed. Some disadvantage to increasing in population continuously. It is difficult to feed whole population in limited landscape. GMO, GMO plants are used to develop and time. GMOs are so impact on ecosystem. Interference in natural dismisses genetic diversity. Also, non-targeted species. Resistance crop may microbes
Your
and
voice
also
is
weed. The
not
process
clear,
of
ma'am.
GMO organism.
Your voice is not clear.
Okay, sir.
Madam, your voice is not clear. Kindly be audible.
Hello. So is it clear?
Yes, yes, now it's clear.
Okay. Genetic modified organisms are so impact on ecosystem. Interference in natural gene flow dismisses genetic diversity, also target non-target species. Herbicide resistance crop may cause the death of beneficial microbes and also weeds. The process of making GMO organism may also mutation occur which cause disease and disorder. When we make GMOs, people pay very less attention towards the another factor, function which also get affected. If genetically modified organisms are safe, then their chances of survival is equal. So then natural selection not play its role. With the help of natural selection, nature supports survival of fittest, and fittest vary according to its ecosystem. And other, if the gene that are introduced in GMOs are more fitted than their wild relative, then the gene are favored by natural selection, and GMOs faster grow and reproduce and take resources from non-GMOs. Many genes function in the presence of another gene. So when we disturb one gene of an organism, it may impact on many another function also. GMOs in environment management. GMOs bacteria can be manufactured biodegradable non-toxic plastics such as polyhydrobutyrate. GMO animals reduce pollutant such as pigs or omnivorous cannot digest a phosphoterase compound phytic acid present in plant. Transfer of bacterial phytase gene to the pig allow them to digest a large part of phytic acids. Parasites, gene from the bacteria which produce a natural insecticide, is transferred to the plant. It reduces use of chemicals and increase productivity. But after several years later, it found that secondary parasite pesticide increase and it reduced plant productivity. And also the primary pest evolved in nature. Herbicide resistant from Herbicides plant can survive in field treated with a particular herbicide. Herbicide reduce use of chemical, but after some year later, it's sown, weeds evolve and registered to this herbicide. Then farmer have to use more potential herbicides, and in it indirectly increase the use of chemical. It is found that monarch butterfly feed on GMO corn, its mortality rate is decreased and also its growth rate is decreases. Genetic modified animal. Mice are frequently used animal because mice are mammal, small, easy to handle, short lifespan, and not too expensive. But mice are not always the true representation of human. In many cases, mice is not good model for study a gene. In that case, another animals are used, such as rabbit and pigs. Genetic modified animals used as a model for medical study. Genetic modified animals provide organs, cells for human beings. Number of patients in modified don't is increased very slowly. Genetic modified animals used to produce protein. So, animals used to improve the food quality also. Impact on human health. There is two case studies. First case study. The first controversy started when Arpad Pastuki revealed his unpublished result of thickening of gut mucosa in response to GM potato. He conducted two experiment group of rat feed with raw modified GM potato and non-GM potato. It was, but its research was taken by many another institute, and it ended up with a positive comment. And after first study, in the first controversy, a article reporting increase in tumor size in that made Roundup was published in Food and Chemical, French molecular biologist. There was huge pressure of media political industry to author did active traction. Each press conference where they released documentary as
Ma'am, your voice is
as
not
it
clear. Your voice is not clear, ma'am.
Hello.
Hello. Hello. Hello. Please unmute yourself. Miss Unnati, please unmute yourself. Miss Unnati, please unmute yourself. Miss Unnati, please unmute yourself.
Unnati, please unmute. Unmute your mic, please. Okay, Siddharth, so stop her presentation and proceed call for next one.
Next participant, Akanksha Kumari. Next participant, Akanksha Kumari.
Yes, sir.
Please present your paper.
Yes, sir.
So is my screen is visible?
Yeah, it's visible.
Okay. Good evening, everyone. Myself Akanksha Kumari from Department of Zoology, Baba Saheb Bhimrao Ambedkar University, Lucknow. My topic is effects of ultraviolet radiation in domestic animals. These are the points we are going to discuss. Let's talk about the introduction. As we all know, solar energy is necessary for all living beings on Earth. Environmental pollution plays important role in global climatic change and that degrade the ozone layer that protects life forms from UV radiations. Domestic animals are constantly directly exposed to the solar
Please
radiation.
rotate your mobile.
Oh, okay, sir. Now is this okay, sir? Is this okay, sir?
Yeah, it's okay. No.
Yes, sir. So domestic animals are constantly exposed to the solar radiation and can cause skin disease, tumor, stress, and even death also. Sensitivity to exposure to solar radiation varies among species to species or even breeds within the same species. Let's talk about the radiation. Radiation is the energy that comes from a source and travel through a space at a speed of light. And this energy has an magnetic field associated with it, and has a wave-like structure, propagation. And we can also call the radiation as the electromagnetic wave. And radiation is classified on the basis of two criteria, by its nature and by its biological effect. By its nature includes electromagnetic radiation, ultraviolet radiation, visible radiation, infrared radiation, and radio frequencies. These are moving from high speed and transport large amount of energies. By its biological effect, it is divided into two parts, ionization radiation and non-ionization radiation. Ionization radiations are high energy radiations, powerful enough to cause displacement of electrons and able to break chemical bonds. Alpha radiation, beta radiation, gamma rays, and X-rays are the example of ionization radiation. Those radiation which are able to alter molecular structures only, but cannot separate electrons from the atom, are known as non-ionization radiation. Example are visible rays and infrareds. Okay. Now we have discussed about ultraviolet lights. Ultraviolet light have a wavelength between 100 nanometer to 400 nanometer, as we can see in this picture, and it is a shorter than visible light and longer than X-rays. It is present in the sunlight. It contributes 10% of the total light from the sun. And type of the UV lights. UV lights are divided into three parts: UVA, UVB, and UVC. And the wavelength of UVA is 350 nanometer to 400, and it is not absorbable in the ozone layer. And it is present more than 98% in UV rays. Okay. And it can cause skin cancer, aging, wrinkle, and eye diseases. Okay. UVB is a 280 nanometer to 215, and it is mostly absorbed and less than 2% in present in UV lights. And it can also cause skin cancer, melanoma, aging, wrinkle, contracts. UVC 100 to 280 nanometers. It is not completely absorbed in an ozone layer. It is present in negligible amount and not present in our earth. These things we are discussed. And now talk about the animal skin physiology. Skin is the largest organ of the body. It is complex structure. Its important function is to homeostasis in our body. It controls the body temperature and also protect our body, animal's body, from physical damage. Also protect against radiation and mainly solar radiation of different wavelengths. Animal skin histology. The layers of skin is divided into three parts: epidermis, dermis, and hypodermis. As we can see in this picture, epidermis, it is a keratinized layer of epithelium. It is a superficial layer of the skin, provide barrier to protection from the invasion of a substance into the body. And now dermis it provides mechanical support and nourishing supplier of the epidermis. In the inner part, it is attached to the hypodermis. It is further divided into two layers: papillary epidermis and reticular epidermis. Papillary epidermis just below the epidermis. It involves loose connective tissue and with abundant blood capillary and provide nourishment to the epidermis. It is not vascularized. Reticular epidermis. It is located under the papillary dermis, and hair rectus muscles is also found in it. And it allows hair to stand erect. And when it is cold and or as a consequences of the animal behavior. Hypodermis is a deeper layer of the skin. Its thickness depends on a skin area of the body and feeding state of the animals. It contains large amount of fat cells. Hairs are the epidermal formation that is most animals are present over entire body surface, and hairs has a root, stem, tips, and which is protruded from the skin. Now we are talking about adaptation. Adaptations of the skin to environmental condition involves morphological particular of the skin and its ability to allow thermal adjustment to environmental variables and thus regulates organisms temperature. Analysis of a different micro and microscopic skin structure in Holstein and Jebo cow found in Jebo to a better adaptation to high temperature. This breed is a shorter, thick hair and greater overall skin thickness is with thinner epidermis and the deeper reticular dermis. Now I'm going to conclude my topic. Domestic animals are almost constantly exposed to the ultraviolet radiation, but change in a climate may increase UVB radiation exposure with possible negative health issues. Sensitivity to the exposure, as I say earlier, is varies from species to species and even between the breeds within the same species. This is my reference. Thank you.
What is the unit of the radiation? How to measure radiation?
Yes, sir.
How
Sorry,
to measure
nano.
radiation? What is the unit of the radiation?
Yes, sir. Hello.
Yeah, I asked ki, what is the unit of the radiation? How to measure radiation?
Sir, we measure radiations in wavelength of nanometer.
No, अगर कि स्किन में रेडिएशन लगी है तो हाउ मच रेडिएशन? हाउ टू मेजर द क्वांटिटी ऑफ द रेडिएशन?
Sorry, sir, I didn't understand.
Okay, no issue. Next participant, Akanksha Burma. Next participant, Akanksha Burma. After that, Chahat Sharma. Please be ready for next participant, Chahat Sharma.
So is my screen visible?
No, it's not visible.
Okay. Good afternoon, everyone. Myself, Akanksha Verma. I'm doing MSc Zoology from Babasaheb Bhimrao Ambedkar University. My topic for presentation today is identification of chromosomal abnormalities with the help of karyotyping. Cytogenetics is a branch of science that deals with the study of genes and its inheritance. Karyotyping is one of the techniques used in this branch. Chromosomes that are thread-like structures located inside the nucleus of animals and plant cells came from Greek word chroma, means color, and soma means body. Animals and plants have designated number of chromosomes present in them. Humans, for example, have 23 pairs, where 22 pairs of chromosomes are numbered, also known as autosomes, and one pair is sex chromosomes. Fruit fly having 4 pairs of chromosome, rice having 12, and dogs having 39. On the basis of location of the centromere, they are of 4 types: metacentric, submetacentric, acrocentric, and telocentric. What is karyotyping? Karyotyping is a process of pairing and ordering all the chromosomes of an organism. This is the most common technique in cytogenetics. Karyotypes can reveal changes in chromosome number, such as Down syndrome, having Down syndrome, also known as trisomy 21. Karyotypes can also reveal more subtle structural changes, such as chromosomal deletions, duplications, translocations, and inversions. This technique checks the chromosome in our cell to see whether we have full set of 46 chromosomes or not. This is a picture of males, 46 chromosomes of males, where sex chromosomes are X and Y, while in females, sex chromosomes are one pair of X and X chromosomes. Cytogenetic testing. This testing is used to help diagnose and classification of leukemia, estimation of prognosis, and evaluation of appropriate treatment regime. To obtain this view of an individual's karyotype, cytogenesis photographs the chromosome and then cut and paste each chromosome into a chart. This is known as karyogram, also known as ideogram. To observe individual's karyotype, a person's white blood cells are first collected from the blood samples or any other tissue. In the laboratory, they are isolated. These cells are isolated and stimulated to begin actively dividing. A chemical called coccicine is then applied to the cell to arrest condensed chromosomes in metaphase. Chromosomes in metaphase are well represented, hence used for this karyotyping. Cells are then made to swell using hypertonic solutions so that chromosomes spread apart. Sample is preserved in fixative and applied to the slide. Geneticist then stains the chromosome with the help of dye. This is to better visualize them and pair them accordingly. The common stain choice is Giemsa stain. There are four types of banding that are in. There are four types of chromosomal banding: G banding, Q banding, R banding, and C banding. Chromosomal abnormalities. These abnormalities happens due to the changes in chromosomes. They may be of numerical or structural type. Numerical type, for example, Down syndrome, which is identified by the third copy of chromosome 21, having 3 copies of chromosome 21, the person is said to be born with Down syndrome. Turner syndrome, which is characterized by the presence of only one X chromosome in a woman instead of normal 2. There are structural type of abnormalities, including deletion, duplication of chromosome, translocation of chromosome, and inversion. For example, Jacobson syndrome, which involves distinct facial features as well as heart and bleeding defects, is identified by deletion on chromosome 11. Sorry. Karyotype can pinpoint wait, my screen is not showing.
No, your screen is not showing now.
So is it
Yes,
visible?
carry on.
Okay. Karyotype can pinpoint translocations, occurs when segment is genetic material breaks from one chromosome and reattaches to the another chromosome. This is called translocation. It can reattach itself on a different part of same chromosome or to the another chromosome. Translocation are implicated in certain cancers, including chronic myelogenous leukemia. Normal result of chromosomes in humans, females having 44 autosomes and 2 sex chromosomes, written as 46, XX, and in males 44 autosomes and 2 sex chromosome written as 46, XY. There are some abnormalities, if you can see in the photo. Down syndrome. In this, it is also known as trisomy 21, as I said earlier. It has extra chromosome 21, Edwards syndrome, trisomy 18, extra chromosome 18, Cuthouse syndrome, trisomy 13, extra chromosome 13, Turner syndrome, monosomy X, single X chromosome in females, triple X syndrome, 3X chromosomes in females, XSY syndrome, 2 Y chromosomes in males. That is all. Thank you.
Thank you, Akansha. Next participant, Chahat Sharma.
Am I audible? Good afternoon.
Yes, audible.
Sir, there is not any option of sharing my screen. Can you please help me?
Okay, send me your presentation in my mail ID.
Sir, is it visible?
Yeah, it's visible now.
Good afternoon, everyone. Myself, Charles Sharma. I'm a research scholar at Maharishi Markandeeshwar Team to be University, Mulana, Haryana, and I'm working on the pesticide degradation using microbial method. So today I'm presenting a poster on the ecotoxicology of cypermethrin and its bioremediation using microbial method. So basically, cypermethrin is an insecticide which is mostly used in the fields of Punjab and Haryana. It is used in crops as well as in livestock and households. And it has been seen that from last few decades, the use of cypermethrin has been increased tremendously. And it also has been seen that there are some negative effects shown in plants as well as on animals and humans this cypermethrin has imposed. Carcinogenic effect has been seen in aquatic animals as well as on fishes that cypermethrin is carcinogenic to them. So it is very important to degrade this cypermethrin which is present in our soil or entered in our food chain. And there are a number of methods to degrade pesticides, including physical methods as well as chemical methods. But all these methods are quite time consuming as well as they are costly. And the another disadvantages of physical and chemical methods are that they generate some secondary metabolites which are equally harmful to the main component. So we are trying to use some bioremediation method, that is we are using microbes, bacteria isolated from soil to degrade these pesticides so that there should be no harmful effect of this pesticide on our environment. So what we have done in our study is that we have isolated nitrogen fixing bacteria from the cereal soil system of different areas. And after all the confirmatory tests, including preliminary test as well as confirmatory test, we have confirmed that these bacteria are nitrogen fixing in nature. And now we are studying their pesticide degradation ability. So our basic biological approach, which is shown in this picture, that what we have done is we have isolated bacteria and now we are checking its ability to degrade the pesticide, specifically cypermethrin. And what we have done is we try to grow our bacteria in the presence of cypermethrin in minimal salt media. Minimal salt media is basically a media which is carbon and nitrogen free media. So what we have done is we add our pesticide cypermethrin at 100 ppm concentration in minimal media and then inoculate our isolated bacteria. And we have seen that all the bacteria show growth in the presence of cypermethrin at 100 ppm of concentration. These graphs are showing the increase in bacterial growth at 600 nanometer. These are the spectroscopic readings, which shows that all of the bacteria with the passage of time shows increase in their growth in the presence of pesticide cypermethrin. So from here we can assume that these bacteria may degrade our pesticide and using this pesticide as an energy source, and that's why they show growth, because in minimal media there is no carbon source, no nitrogen source. So bacteria is taking energy from this pesticide and show growth. And that further confirmation can be done by using GCMS, which is in process, which results has not been come yet. But this is the basic procedure which shows that we can degrade our pesticide using bacteria in this manner by growing them in a carbon nitrogen free media. And future perspective of this study is that this is a sustainable this helps in sustainable agriculture and also improves environmental health, as well as it also uh, it also help to reduce all the environmental pollution caused by this pesticide. Thank you.
Thank you so much, ma'am. Now, next participant, Nancy Jadhav. Next participant, Nancy Jadhav.
Yes, sir. Good evening, everyone.
Is my screen visible?
Yes, visible.
Okay. I'm going to present about the preliminary investigation of parasitic infestation in the tissues and organs of the Xanthodon cancella. The Astrongylus larvae infects the ovaries of the freshwater fish Xanthodon cancella, and it affects the host population as it influences or reduces their reproductive potential. It also leads to delay in the sexual maturity of the fish. The nematode could be seen in this picture, the bright red color nematode. It belongs to the order Ascaridia and family Dictophymidae. The experiment required materials are the fish specimen to be collected from fresh waters and the collection of the parasite from fish. The living specimen of this fish were collected from the lower lakes of Lucknow, and they were brought to laboratory and examined before morphologically and internally in the occurrence of helminth parasite. The fish specimen was dissected out in the physiological saline that was a sodium chloride solution of concentration 0.75%, and thus the parasite was collected. The parasite that was collected was bright red in color, and its length was 450 to 680 microns, which was found encapsulated or free in the body muscles, swim bladders, liver, intestines, ovaries of the host fish. Its existence or prevalence increases when the pre-spawning and post-spawning period, and majorly it was recorded, its maximum existence was recorded during the month of November and March. The parasite infects females more than males and causes histopathological damage more to the ovaries or oocytes of the fish. The histopathological damage caused by the parasite in the ovaries is characterized by necrosis, reduction in size, and number of oocytes. It also leads to atresia and degeneration of developing oocytes. This has been recorded in 4-month period. The reduction of feudicity and further decline in the population of infected fish has been recorded. The parasite influences the population of the fish due to reducing its reproductive potential, in which the oocytes are highly affected as the yolk of the oocyte gets globular and liquefied, which reduces the formation of the progeny. This is a slide showing the ovaries, normal ovaries, and then the histopathological study that was conducted. It was the infected gonads of the fish was taken out and fixed in the alcoholic Bowen's fluid for 72 hours. After the complete removal of the picric acid, the tissue was processed for preparation of the paraffin wax blocks, and tissue was cut at 6 micron thick section, which after proper processing prepared in permanent slides. The result, or the actual result recorded, was that 43 were found infected out of 103 fish specimen from Astrongylus larvae, and their maximum prevalence was recorded in the month of March and November, and its highest minimum was recorded in the month of September. Thank you.
Thank you so much, Nancy. Now the next participant, Asteria. Next participant, Asteria.
Nancy, am I audible? Nancy. Nancy, unmute yourself. Okay. So
Nancy,
Yes, sir.
What was the sample size, Nancy?
Yes, sir.
What was the sample size of your experiment?
Sample size, sir, it was, wait a minute, sir. Six, 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 6 micron. So 50, there was 103 fishes, and out of 43 were found infected.
But you are showing 100% prevalence in the month of something in your
In the
PPT.
month of November, sir, 100%. In the month of November, which the fishes showed 100% infection.
But in the month of November, how much fish you have observed?
Sir, in the month of November, we observed around 50 fishes.
And everyone having the infection of Euston glidis.
So, yes, sir.
And how you identified Euston glidis?
So the nematode is bright red in color.
Bright red in color means Euston glidis larvae.
Yes, sir. The larvae is bright red in color, and
Why bright red in color?
its size was around 450 micron to 680 micron.
460 micron.
450
open your
to
video,
680 micron.
Just open your video first of all. I'll try to communicate to you. 1 minute. It's just for a sake of interest, not for any other thing. Okay. Open your video, Nancy. Just a minute, Siddhartha.
yes, sir.
Okay. Nancy. Nancy.
Yes, sir.
Yes, you are telling 460 micron.
So I was
Okay.
telling 450 to 680 micron, sir.
Okay, 450 to 600 micron. Okay.
Yes, sir. It was the size of the nematode.
Nematode. What will be the size in millimeter? Unmute yourself, unmute. You are mutated. Unmute first. Unmute yourself. No sound, please. Unmute yourself first. सिद्धार्थ आवाज आ रही है?
No, sir.
कोई बात नहीं सिद्धार्थ, फिर प्रोसीड करिए आगे, हम लोग फिर कभी डिस्कस कर लेंगे. ओके.
Yes, sir. Yes.
A little doubt about the. Thank you.
Ashwarja, please proceed your PPT. Aishwarya, please proceed.
आज भी थी उसकी सुबह और शाम।
हाँ सिद्धार्थ, किसका नंबर है एक बार?
Sir, just connect Aishwarya Sharma.
ऐश्वर्या, ओके।
Actually, sound problem. PPT is visible. It's maybe some sound problem. Next participant, Rabia Hafiz. Next participant,
Yes.
Rabia Hafiz.
Yes, sir. Yes.
Please.
Sir, is this screen available? Visible?
Yes, visible.
Good evening, everyone. Myself, Rabia. I'm pursuing my master degree from Babasaheb Bhimrao Ambedkar University. Today, I'm going to present the topic that is the comparison of genome organization between lower and the higher vertebrates. First of all, what is a genome? Genome was first coined by the Hans Winkler in 1920. A genome is the entire set of DNA instructions which are found in the cell. Genome is like our body's instruction maintenance that contain all the genetic information needed for growth, development, and the functioning of the cell. It is a unique set of instructions that gives a shape to an individual's traits and the characteristics. Genome organization. The linear arrangement of DNA and their partition into chromosome is the genome organization. Organization is the arrangement of DNA sequences inside the nucleus and the dimensional structure of the chromosome. Genome Genomes which are in their native states are folded into complex high structures. Hierarchical folding of DNA give rise to chromatin fibers, chromosomes, domains, and finally chromosomes. But at the lowest, DNA is wrapped around an octamer of core histone proteins, which are the primarily architectural elements of the chromatin fiber to form the nucleosome. As we can see in these pictures, the nucleus containing number of chromosomes. Chromosomes contain the 100 of nucleosomes, and these nucleosomes are joined by the DNA and that run between them. Next one is a comparative genomics. Comparative genomics is an exciting field of research in which scientists use the variety of tools, including computer-based analysis, to compare genes or the complete genome sequence of different species. By comparing a sequence of genome of different organisms, researchers can understand what at the molecular level distinguishes different life forms from each other, which is very extremely important. This field can eventually help us to understand the methods of genes that are associated with certain diseases, which might lead to the production of more factor of medication. It also provides a powerful tool for studying evolutionary changes among organisms, helping to identify genes that are conserved or common among the species. Next, how the genomes are compared. Genomes can be compared according to the gene size, genome number, and the genome shape. We can compare the genomes of the different vertebrates on the basis of the some features like the genome size. Like lower vertebrates have generally smaller genomes as compared to the higher vertebrates which have the larger genome. In the lower vertebrates, there are fewer non-coding regions, but in the higher vertebrates, more extensive non-coding regions. Repeated elements are less complex in the lower vertebrates, but in the higher vertebrates, complex repetitive elements, transposons are present. Regulatory elements like simpler regulatory network is present in the lower vertebrate, but in case of higher vertebrate, elaborate regulatory network facilitating intricate genes. Here in the next slide, a statistically significant inverse correlationship was observed between the mean genome size of each vertebrate class and number of extant species. In the first figure, there are species number and the genome size is in the main tags of vertebrate is differentiated according to the species number and the genome size. In the x-axis, there is species number is mentioned. In the y-axis, the genome size is mentioned. Here, different types of vertebrate classes are differentiated according to their species number and the size. In the second slide, there are genome sizes in amniotes, which are also differentiated according to the species number and the genome size. Third one, in the amniotes. In amniotes, there are also the species number and the genome size is on the basis of species number and the genome size, we can differentiate the different types of amniotes. If only the values of amniotes are included, the correlation was linear, like in the third figure. There is linear correlation, but in case of the amniotes were considered, the correlation was identified by a power-like curve. But there are some similarities are also available in the genome organization. Like lower and higher vertebrates share some fundamental similarities in genome organization, including the presence of genes, coding regions, and the regulatory elements. As we can say that the zebrafish has total of 25 pair of chromosomes, and it has more than 1 million base pairs that are about half that number of in comparison with the most of mammalian genomes that ranges from roughly 2 to 3 and more billion of base pairs. The genome of Commodus, which is the largest lizard living today, it has some show similarities with the human genome, like 20 pairs of chromosomes present in the Commodus lizard, which are total 40 chromosomes. Like in human, there are 23 pairs of chromosomes, which are 46, almost like a similar chromosome number in their two classes, like a lizard and the human. Birds and mammals also show similarities in their genome organization. They both have the same DNA composition, like the nitrogen bases are same in their in the DNA composition, like adenine, guanine, cytosine, and the thymine. This completed G-fish genome will be an essential resource that derives the study of gene function and the disease in people. In this slide, the picture, ortholog genes share between the zebrafish, human mouse, and the chicken genome. And the human mouse and chicken and the zebrafish have almost the same genome. As conclusion, genome comparison provides insights into genetic relationship, evolution, and the disease susceptibility. Analyzing similarities and differences helps researchers understand genetic diversity and identify key genes for various traits. Scientists can better understand the appearance and behavior and biology and biochemistry of different organisms by studying genomics. These are my references. Thank you so much.
Thank you, Ravia. Next participant, Ankit Kumar.
Yes, sir.
Please present your paper.
Hello.
Yes, sir.
Sir, is it audible now?
Yeah, it's audible now.
Sir, it is
Please
visible,
present.
Ankit Kumar.
Yes. Please present. After that, Asoja Sharma will be present.
Good evening, sir. Sir, my name is Ankit Kumar. I am doing MSc last year in Babasaheb Bhimrao Ambedkar University, Lucknow. Sir, I am presenting my topic, role reversal of histone proteins in gene regulation. Next,
Ankit Kumar, please wait. Asoja Sharma, please ensure your screen now.
Yes, sir, already shared. Is it visible?
Yeah, it's visible, but please unshare it now.
Aishwarya, please stop sharing so that I can present, and thereafter you have to.
Okay, sir.
Yeah. You can share your slide, please.
Ji, sir. Thank you, sir. Today I am talking about the role reversal. Slide, sir, is visible?
No, it's not visible.
Sir, something happened, sir. I am doing. इस पर जाएंगे.
Aishwarya, stop sharing. Your screen is showing here.
Sir, I have stopped it.
Click on once more on the share so that it, yeah, okay. Now Ankit share.
Sir, I am doing but not happen.
Yeah, it is now working. Okay.
Sir.
Ankit Kumar, please present.
Sir, 1 minute, sir. I am doing. Oh. Sir, can you help me, sir? I am clicking on 3 dots and then option coming share, but
You can stop the sharing, then start the sharing.
sir, it is visible.
Yeah, it's now visible.
Sir, thank you, sir. Today I am talking about the role reversal of histone protein in gene regulation. First of all, I am talking about what is gene regulation. Our each cell having the same copy of DNA, but every cell functioning and morphologically different due to the processing mechanism of gene regulation. These are our content introduction. Histone modifications, likewise DNA, like introduction. Histone protein. What is histone protein? The most abundant protein in chromatin are the histone, which are small positive charge proteins of 5 major type: H1, H2B, H2A, H2B, H23, and H24. H1, H2A, H2B, H3, and H4. All histones have a high percentage of arginine and lysine positive charge amino acid. They share a net positive charge. These positive charge attract the negative charge on the phosphate of DNA. This attract hold the DNA in contact with the histone proteins. Each of histone proteins that make up the nucleocore particle has a flexible tail containing from 11 to 37 amino acids which extend out from nucleosome. Nucleosome is a complex structure. It is a repeating unit of chromatin. Positive charge amino acid in the tail of the histone interact with the negative charge of phosphate of DNA, keeping the DNA and histone tightly associated. The DNA is present in form of the chromatin in a chromosome. The chromatin is made up of DNA and histone and non-histone protein. This is a form of supercoiling DNA. The repeating unit of chromatin is nucleosome. The nucleosome is a core particle consisting of DNA wrapped about 2 times around an octamer of 8 histone proteins, 2 copy each of H2A, H2B, H3, and H4. The fifth one H1 is act as a linker, which is act as a clump, join the DNA and existing the DNA from nucleosome. The DNA is direct contact with the histone octamer is between 145 to 147 base pair in length. Histone modifications, histone acetylation. The histone acetylation occur at the amino terminal tail of these histone by an enzyme called histone acetyltransferase HAT. The acetylation of histone leads the uncoiling of this chromatin structure, and this allow it the assist by the transcriptional machinery for expression of gene. It is a produce euchromatin, which is less dense and transcriptionally active. This chromatin are occur in chromosome arm P and Q arm. This is a major material of chromosome which undergo the normal process of condensation and decondensation in the cell cycle. Euchromatin constitutes the majority of chromosome material and is where most transcription takes place. And second is the histone deacetylation. The both histone acetylation and deacetylation are reversible modifications. Histone acetylation is balanced by the another enzyme that remove these methyl acetyl group. Sorry, this is mistake. Methyl group, not this is acetyl group, which is called histone deacetylase. HDAC. Histone deacetylase is produced dense and transcriptionally inactive chromatin that called heterochromatin. Heterochromatin are present on the centromere and the telomere region. The heterochromatin present in all chromosome in permanent stage, and Y chromosome also consists of large of heterochromatin. This is called constitutive heterochromatin. Both the histone acetylation and deacetylation refers as epigenetic regulation. It is inheritable from one generation to another generation. DNA methylation. DNA methylation is refers the addition of methyl group at the CpG island. CpG island, that is the pairs of C and G. C means cytosine, DNA dinucleotide. And at this region, the methylation is occur. The DNA methylation stabilizes, alters the expression of gene. So it occurs when cell division and cell differentiation from embryonic sensor to specific tissue. So it is essential for normal development and associate with the other process such as genomic imprinting, X chromosome inactivation, etc. The abnormal DNA methylation is implicated carcinogenesis or development of cancer. So, normal function of DNA methylation is critical regulatory mechanism for our cells. The DNA methylation is altered transcriptional gene in two ways. The first is the DNA methylation cause the physically impede the binding the transcriptional protein, and second is methylated DNA may be bound by the protein known as MBD or methyl CpG binding domain protein. The MBDs require additional protein like non-histone protein and other regulatory protein. This modifies the histone forming condense inactive heterochromatin, that is inactive, basically transcription is silence. And these are my conclusion of modifications. histone modifications. The histone modification protein include the addition of methyl group, acetyl group, phosphate, and ubiquitin altered chromatin structure. Some of these modifications are passed to daughter cells during cell division and to future cells. These modifications leads directly expression of genes such as embryonic sensors to specific tissue as well as genome imprinting and X chromosome inactivation. The DNA acetylation and deacetylation to allow transcription machinery to gene switch on and switch off. As a result, euchromatin and heterochromatin are formed. These are my references. Thank you, sir. And thank you to all organize this conference and give me opportunity for the participate. Thank you, sir.
Thank you, Ankit. Next participant, Salima Batol. Next participant, Salima Batol.
Yes, sir. Wait a minute, sir. Sir, my screen is visible?
Yeah, it's visible.
Good afternoon, sir. Good afternoon, everyone. My name is Salima Batul. From Department of Zoology, Baba Saheb Bhimrao Ambedkar University. My review article on inside structural genomics. First, introduction. The term structural genomics typically refers to the collective or comprehensive set of three-dimensional structure of biomolecules, primarily protein within an organism. It describes also 3D structure of every protein encoded by a given genome. In structural genomics, one of the most powerful
Your PPT is not visible. Please open the PPT.
Okay, sir, I'll try. Sir, now it's visible?
No, no, no.
Sorry, sir, there will be an issue to open the PPT. I'll try.
Okay, please try. Next participant, Sharja Sharma.
Hello.
Yes, please present.
Shall I start?
Yes, please start.
Hello.
Hello.
Shall I start?
Yes, please start.
Myself, Aishwarya Sharma from Career Point University, Hamirpur. And topic for my presentation is harmful consequences of microplastics on flora and fauna, a review. So coming over to introduction, microplastic pose severe threats to flora and fauna. These are minute plastic particles resulting from the breakdown of larger plastic debris, and they are known to contaminate ecosystems by harming plant life as well as fauna. Ingestion of microplastics can lead to reproductive issues, physiological damage, and ecological imbalances, endangering the diverse ecosystems. In aquatic environments as well as in terrestrial environments, they are known to cause harmful consequences. Now, coming over to the contents, what are microplastics? How do they enter into our ecosystem? How they affect flora? How they affect fauna? What can be done with context to this area? And conclusion. So first of all, what are microplastics? Microplastics, these are minute plastic particles which are measuring less than 5 mm. They pose a pervasive threat to flora and fauna in terrestrial as well as aquatic ecosystems. These particles infiltrate soil and water bodies and also disrupt many cycles and processes, which eventually lead to the imbalance in ecosystem. And once in environment, microplastic can be transported by winds, water currents, wildlife, and they can also pass on through the food chains. So this is the matter of great concern these days. Next, how do they enter into ecosystem? So fragmentation of larger plastics, because it usually takes a longer durations for their biodegradation. Then synthetic fibers are there, industrial processes, improper waste disposal. So these are certain practices which can cause the mitigation of the microplastics. Next, how they affect flora. So when they enter into the terrestrial ecosystem, they contaminate soil, which eventually give rise to the base of plants, and which can be transferred to high trophic levels through food chain. These also impact ecosystem, and water retention and drainage is also a cause. Then as the roots absorb nutrients and transports them, so likewise the microplastics along with the processes get into the fruits or the product, which then is being taken by other animals. And next, this is again a little description about that how they accumulate in soil and then the broader ecosystem. They include potential harm to organisms that interact with plants like soil dwelling invertebrates and microorganisms. Disruptions to these components of ecosystem can have cascading effects on plant health. Microplastics also contain various additives and chemicals that may leach into the soil, and then again resulting to toxicity in case of plants and animals. Next,
coming over to how they affect fauna. So, fauna are generally the animals which live in aquatic as well as terrestrial ecosystems. So, through ingestion, as fauna may mistakenly consume microplastics either directly or indirectly. In aquatic environments, filter-feeding animals like zooplanktons and some shellfish can ingest microplastics while feeding on particles. And then when these small organisms are taken up by the larger ones, so the microplastics are transferred to them. Next is the physical damage. So they can also cause physical damage to organisms that ingest them. The particles may accumulate in digestive tract, potentially causing blockages, inflammation, and other internal injuries. And this case is very common in case of humans these days, as we are consuming water from plastic bottles, the packed water bottles likewise. So that is also uh, contributing into such microplastic blockages and all. Then next, and also serving as a cause for cancer. Next are chemical exposure. So microplastics can absorb and accumulate chemical pollutants from the surrounding environment. When organisms ingest microplastics, they may also be exposed to these pollutants, which can include harmful substances like pesticides, heavy metals, and endocrine disrupting chemicals. This can lead to toxic effects affecting the other systems like immune system or reproductive behavior in case of fauna. Next, are bioaccumulation, behavioral changes, and habitat alteration. So in bioaccumulation, microplastics can enter food chains with smaller organisms, as I already explained, and then this will lead to bioaccumulation, as we also study in case of that ppm levels get increased. So likewise, also the microplastics can cause more harm in higher trophic levels, posing greater risk to the top predators and consumed by the humans. Then coming on to the behavioral changes, so some studies suggest that exposure to microplastic may induce behavioral changes in fauna. For example, fish exposed to microplastic may exhibit altered feeding behaviors, reduced exploration, or changes in the reproductive activities. And these changes can have the effects on ecosystem and the survival and fitness of affected species, and many of the people are also taking this seafood like fish and all. So microplastic can get transferred from one level to another. Then next is habitat alteration. So accumulation of microplastics in sediments and soils can lead to changes in habitat structure and composition. This can affect the fauna and rely on these habitats for shelter, breeding, and feeding. So now, what can be done with context to this area? If talking about research, then a lot of work is going on this topic related to microplastic, but still a lot is to be done, because as population is increasing, pollution is also increasing, and waste management like issues are of major concern. So I think that this is a burning topic, and more is we can do more. So it's important to note that the effects of microplastic on flora is an active area of study, and our understanding of these impacts is continually evolving. The widespread presence of microplastic in environment underscores the importance of finding sustainable solutions to reduce plastic pollution and minimize its impact on ecosystems. Mitigating the impact of microplastics on fauna requires concentrated efforts to reduce plastic pollution at its source, improve waste management practices, and develop technologies to remove existing microplastic from the environment. Additionally, research on long-term effects of microplastic on different species and ecosystem is crucial for developing effective conservation and management strategies. So as a conclusion, microplastics pose severe threats to flora and fauna. These tiny particles resulting from plastic degradation disrupt ecosystem by contaminating soil and water. Ingestion by organisms lead to adverse health effects, bioaccumulation, and ecosystem imbalance. Urgent action is crucial to mitigate this pervasive environmental hazard and protect biodiversity. The widespread presence of these persistent pollutants emphasizes the urgent need for comprehensive mitigation strategies to safeguard the delicate balance of ecosystems and ensure the well-being of diverse plant and animal species. Thank you.
Thank you, Asurya. Next participant, Salima Batol.
Yes, sir.
Are you ready now?
Yes, sir. I will try to connect this PPT. 1 minute. Sir, is it visible?
Yeah, it's visible.
Sir, PPT is visible?
No, PPT is not visible. First open the PPT. Yeah, now visible.
Okay, sir. Hello, sir. Good afternoon. Sir, good afternoon, everyone. My name is Salima Batul. I am Salima Batul, Department of Zoology from Baba Saheb Bhimrao Ambedkar University. My review topic on inside structural genomics. First, introduction. The term structural genomics typically refers to the collective or comprehensive set of three-dimensional structures of biomolecules, primarily protein within an organism. It also describes 3D structure of every protein encoded by a given genome. Structural genomics, one of the most powerful approach for defining the structure of protein. There are various experimental techniques through which we determine that particular protein structure are X-ray crystallography, nuclear magnetic resonance, spectroscopy, and cryo-electronic microscopy, as well as computational method for structure prediction and modeling. An overview of this structural genome approach adopted to illustrate three-dimensional structure of protein are shown in figure. This figure shown the genome of a particular organism, of a particular cells or organism. Of that genome, the protein structure are resolved using high throughput crystallography or NMR technique, which are then deposited in protein data bank or there are computationally predicted using homology modeling software. Also, that state protein structure can be further used for drug development studies, function prediction, and metabolic pathway analysis. Methods. There are various methods to determine that particular protein that embedded on the structure, embedded on the genome. So there are various methods. There is through experimental method or sequential method. There are high resolution structure they give of that particular protein. We also determine these two methods, a particular protein structure and their function. And the another method is also threading method and ab initio methods. And this is also a low resolution structure method. And so we also determine particular protein structure through these two methods also. Up now to study that different review, different research literature or review different study of different literature. Now the question: structural genomics in biomolecular drug design. What are the role? What are the impact on what are the impact of structural genomics in biomolecular drug design? There are role of structural genomics in biomolecular drug design in medicine for several reasons. First is target identification, means knowing the structure of a specific protein involved in disease help to identify potential target for drug development. If a protein is responsible for a disease, understanding its structure can aid in drug designing that specifically interact with it to modulate its activity. And another structural genomics role is also rational drug design. Once the structure of target protein is known, scientists can use computational method to design molecules that fit into active site of that particular protein, either enhancing or inhibiting that function. Means a particular protein we find according to the structural genome by X-ray crystallography or another computational method. So we find that particular protein structure. After that, various scientists active site of that particular protein also make that inhibitor. मान लेते हैं कि वो प्रोटीन जो है एक वायरस का हो या एनी अदर डिजीज काजिंग पैथोजेंस का हो, तो दैट पर्टिकुलर प्रोटीन का डिटर्मिनेशन करके हम उसका इनहिबिटर ढूंढ के दैट पर्टिकुलर डिजीज जो है वो हम डिफाइन भी कर सकते हैं. दैट आल्सो भी कंट्रोलिंग. Now the another role is drug repurposing. For knowledge of this protein structure, about हमें इस protein structure के बारे में knowledge होने के बाद allow the scientists to repurpose existing drug for new uses. Also understand how a drug interact with multiple proteins can lead to its application in treating different diseases. Another role in vaccine development. Structural genomics contribute to the design and development of vaccine by providing insight into the structure of antigen and how they interact with the immune system. This understanding helps in creating more effective vaccines. Also, the another structural genomics role in biomolecular drug design is personalized medicine is also understanding the structural variation in proteins among individuals can also aid in personalized medicine. So these are some few structural genomics role in biomolecular drug design. There are several uses also. Now the examples of structural genomics. For example, two examples I discuss here. That is the Thermotogo maritima, thermophilic bacteria, and Mycobacterium tuberculosis. The Thermotogo maritima is a thermophilic bacteria. That protein structure is defined the protein structure initiation to solve this structure of all protein in Thermotogo maritima, a thermophilic bacteria. That Thermotogo maritima was selected as a structural genomics target based on its relatively small genome, consisted of 1,877 genes, and the hypothesis that protein expressed by a thermophilic bacteria would be easier to crystallize, so they would be selected. Mycobacterium tuberculosis is also an example of that structural genomics. So the goal of TB structural genomics consortium is to determine the structure of potential drug target in Mycobacterium tuberculosis, the bacterium that causes tuberculosis. So the development of that novel drug therapies against the tuberculosis are particularly important given the growing problem of multi-drug resistant tuberculosis. Now conclusion. In the conclusion, structural genomics have significant advance our comprehension of biological system at molecular level. Also, it is an impact on drug design, have been transformative, enable the development of targeted and efficient therapeutics. Continue advancement in techniques and technologies within the structural genomics promise further breakthrough the way for more precise and personalized medicine in future. These are the references. Thank you, sir. Thanks so much.
Thank you, Salima.
Yes.
Now, next participant, Ravi Kumar. Next participant, Ravi Kumar, BVAU, Lucknow. Next participant, Ravi Kumar. Please unmute yourself. Hello, Ravi Kumar. Ravi Kumar. Ravi Kumar, please present your paper. Next participant Pankaj, Yasika, Kamtenal, Monali, and Nikesh. Anyone here? Hello, hello everyone. Today the session will be closed now. Then we will again meet tomorrow morning 8:45 o'clock. The oral and poster presentation session will be start, and the guest speaker session will start again 10 o'clock. And tomorrow's guest speaker is Dr. Gaurav Saxena, Dr. Nikul Chavada, Dr. Sanjay De, Dr. Hitesh Kumar Solanki, and many more. So goodbye.